Hello again, everyone. Welcome in to the stream. Hello, hello. We are we are doing. What are we doing today? I don't actually. No plan today. We're just freestyling the study session. Um, so we'll see what comes of it. I think I'll do a little bit of finish off what I was doing yesterday. Oh, well, not finish off because we finished off the like skills sort of uh 65 question base skills thing. And I got about half of those right, so definitely need work on that. But just because I did a lot yesterday, I'm not really going to go back to that. So I might watch a few of those tutorials that I was doing or that I was planning on doing yesterday, and then I might go into some. I think I'll go into some actual questions because I'm kind of itching to, you know, do that because we haven't done it for a while. So that's kind of the rough plan, and we'll just kind of freestyle it a little bit from there. So this is the test I'll be using for the practice questions. GAMSAT practice test A, the pink one. So yeah, so like I said, 52% in that. So I guess we'll need to work on that a little bit and try and get that to 100% at some stage. I struggled with the logarithms and what else? Some of the index laws and some fraction stuff. Some of the algebraic manipulation stuff as well. So I don't know. We'll come back to that at some stage, but maybe we'll just jump into this one for a little bit. That's got a lot of content. It's pretty crazy.
Welcome to your Section 3 Science Crash Course. So in this course, I don't really intend for it to be a rehash of a science degree or something like Khan Academy, because really what we want to be doing in Section 3 is demonstrating depth of understanding of relatively simple concepts. So each of these areas is going to be the core things that you really need to understand well, but you'll see that there's not that much detail. So if you want to supplement it with more detail, that's fine. I don't think that's necessary, but I do understand that some people really like adding in more detail so they understand it better. So as long as you're doing something to understand the ideas presented here better, that's
Oh,
okay.
whoops.
But I don't want you going down rabbit holes trying to accumulate more and more information, because generally the questions which are examining these concepts are more about the problem solving and reasoning skills that you demonstrate, not the knowledge. So you'll find that these simple ideas actually cover a vast number of questions, and it's really how you apply them to problems. So don't go too deep down rabbit holes, but this will be a really good kind of refresher covering all of the major science knowledge that you need to know for Section 3. I've seen it many times that people have actually known more niche little facts about these concepts, but don't have that in-depth understanding, and that's what's leading them astray. So it's very, very rare that you're going to be asked to recall some niche bit of scientific knowledge. It's usually all there in front of you, and it's how you relate that complicated information to your simple understanding to solve a problem. So just to emphasize that I really think the important thing is understanding these core concepts well and not delving too far into the content. Okay, so let's dive in with some cell biology. Now, to keep it simple, really, the purpose of every cell is to produce proteins. And that's because proteins really are the functional unit of everything. So, all of the reactions that occur in our body are mediated by proteins. All of the structures, so the skin, our eyes, our hair, whatever, it's all formed by protein. So, really, what I want you to just remember is the goal of everything, every cell, is to produce proteins that allows the cell to do its function. And by doing its function, it allows the organism, like us, to survive. So we have billions of these cells making proteins, allowing them to do their function. Now, how we do that is we need to make the proteins, right? So just to introduce you into cell biology, we're just going to go through how we actually make these proteins. And that'll also introduce you to a few organelles, which are just parts of a cell. So these are parts of the cell that have functions in making proteins. So firstly, we have the nucleus, and in the nucleus, we're housing our DNA. So you've probably heard of this, and that's basically the DNA is the code for all of the proteins in the body. So I like to think of it like a book. So I've drawn a really bad book here, and it's D for double-stranded. So it's this double-stranded molecule. So you can think of two strands and they're matched up by different letters and they're double-stranded. And I just like to think of it as a book, and we want to keep that book safe. And we also don't want to, every time we make a protein, we don't want to replicate the entire book because it's going to take a lot of energy. We don't necessarily need all those proteins. So if you think about the stomach, we want to be making different proteins to, let's say, our skin, okay, because they have different functions. So depending on the cell, you're going to be only making certain proteins. So you don't want to just be making the whole book all the time. Okay, so the nucleus is really there to keep the DNA safe, and then when we want to make a protein, we just use the parts of that book that we need. So let's say we want this page that I've opened it to. Think of it like you photocopy it. Okay, so you photocopy just that page of that book, so you've just got what you need. Now, the other really cool thing here, so we've gone from double-stranded to single-stranded. I'll just mention that. So we're using like a page as an analogy, but it's just like a single-stranded thing of letters, which we'll go into in a second. But we've got this page, and it's got all these words on it. But we can actually go even further than that. So if we wanted to, we could actually just get the top part and the bottom part and join these together and just kind of cut out that middle part. Now, how we cut out the part we don't want to merge the parts that we do is through proteins. So just to give you one idea of the kind of functions of them. But this is how we get so many different proteins from our DNA sequence. So I think there's something out there like we've got roughly the same amount of genetic material as a tomato, but the variance of the genes that we have is why we have so many more proteins than a tomato. And that's through this technique of just taking the bits that we need. So let's say we take those bits that we need. This is now called our RNA strand, specifically our mRNA strand. M just means messenger, or you can think of it as message. So it's just carrying the message, and this is single-stranded. So everything apart from the DNA is going to be single-stranded. This RNA then goes to the ribosome, and really what the ribosome does is it just holds this strand. So you think of the ribosome.
Do I take notes of this? Do I take notes? Do I take notes? Do I take notes? I think we do. I think we do. I kind of know this stuff a little bit, but I mean, I couldn't give a synopsis of it like old mate is doing, so I guess we take notes. I guess we take notes. Bank, knowledge bank. It's alright. I think that's what he said.
Kind of as this structure, the strand just comes in, and the ribosome just this strand, okay, specifically taking the bits that we need. So let's say we take those bits that we need. This is now called our RNA strand, specifically our mRNA strand. M just means messenger, or you can think of it as message. So it's just carrying the message, and this is single-stranded. So everything apart from the DNA is going to be single-stranded. This RNA then goes to the ribosome, and really what the ribosome does is it just holds this strand. So you think of the ribosome kind of as this structure, the strand just comes in, and the ribosome just kind of sits here like this. This is how I think of it anyway, and it just holds that strand there. So just imagine it holding it. And it has these letters, and again, I'll go into what the letters are a bit more later, but you might have things like these 3 letters, and it just holds that there. And then what happens is this. Now, how we cut out the part we don't want to merge the parts that we do is through proteins. So just to give you one idea of the kind of functions of them. But this is how we get so many different proteins from our DNA sequence. So I think there's something out there like we've got roughly the same amount of genetic material as a tomato, but the variance of the genes that we have is why we have so many more proteins in a tomato. And that's through this technique of just taking the bits that we need. So let's say we take those bits that we need. This is now called our RNA strand, specifically our mRNA strand. M just means messenger, or you can think of it as message. So it's just carrying the message, and this is single-stranded. So everything apart from the DNA is going to be single-stranded. This RNA then goes to the ribosome, and really what the ribosome does is it just holds this strand. So you think of the ribosome kind of as this structure, the strand just comes in, and the ribosome just kind of sits here like this. This is how I think of it anyway, and it just holds that strand there. So just imagine it holding it. And it has these letters, and again, I'll go into what the letters are a bit more later, but you might have things like these 3 letters, and it just holds that there. And then what happens is this other molecule comes with the opposite strand. So again, I'll go into the matching in a sec, but this comes along and binds to it. So the ribosome really is just there to hold the strand and hold this thing that matches those letters on that strand together. Now, attached to this molecule that comes across and binds is this thing called, running out of colours, is this thing called an amino acid. So an amino acid, okay? And basically, once that amino acid has come across, so we've got 1 amino acid per transfer molecule, and that brings a very specific amino acid there. So that's why we have this matching of sequences, so that we're only getting the amino acid that we want at that point, okay? And then we just kind of wash, rinse, and repeat. So we just go through and we just read this strand 3 pairs at a time, and each time the ribosome is just stabilizing the strand, allowing the transfer molecule to come over and bind, and then also stabilizing the amino acid that it brings. So over time, you just get this longer and longer chain of amino acids based on what the 3 letters are that are in that message strand. So imagine you have 20 letters. You read the first 3, the second 3, the third 3, and so forth until you reach the end of the chain. And then you've got this amino acid chain. That amino acid chain then goes to this thing called the Golgi apparatus, and through that process, it kind of modifies it. You don't really need to know much more than that, but just modifies it and packages it. And then through the different amino acids that you have, that protein will fold in a different way. So all these different colors are either attracted or through the different amino acids that you have, that protein will fold in a different way. So all these different colors are either attracted or repulsed by each other, and that will allow it to fold in a different way. And the way that it folds dictates its function. So if we have this configuration of amino acids, maybe it folds like that. Okay, so it's a really complicated. folding process. You don't really need to know it. Just know that the configuration of amino acids will determine the way it folds, and that will determine its function. So just to recap that process, we have the nucleus, which houses our DNA, which is like that book. We then want to produce a copy of a part of that book. So we copy a page, let's say we photocopy a page, and we leave the book nice and safe in the nucleus. We then use that page and we cut out the bits of the page which we don't want, and that becomes our mRNA strand. And that gets read systematically in pairs of 3 by the ribosome. And how it does that
The
is it
idea
reads
of
the 3
structure
and it
and
brings across another
function is
molecule
really
which binds to
interesting.
those exact 3 pairs, and that molecule itself has an amino acid. So by matching those 3 pairs, we're getting just the amino acid
Like
that we
you
want.
wouldn't
And then as we
cook
keep reading that
a
strand and
meal in a
repeating this process,
hallway.
we add more and more amino acid molecules to our chain. That chain then gets modified and it folds together based on the amino acids which are in it, and that produces our final protein molecule, which can then do various functions. And really, just to touch on the last thing, is the cell membrane. This is just a protective layer around that cell. Alrighty, so in the last video, we kind of went through how you go from DNA to RNA to proteins. I'm just going to show you a little bit more detail about those letters that I was talking about, so the code that makes them up. So in DNA, we have 4 letters. There's A, G, C, and T. And it's not really that important what they are.
I kind of know like the basics of this stuff, I guess. So I'm not gonna spend much time on this, I don't think. Maybe chemistry would be good. Yeah, maybe chemistry and physics would be good. Is there a math one? There is.
Okay, so we're going to get on to chemistry now, and we're just going to start with some basic stuff about atoms. So what I've drawn here is a nucleus, and in the same thing as a cell, really. It's just the center thing which houses some important stuff. Except in this case, our nucleus of the atom is actually the thing which has things called protons and neutrons. So protons are positively charged and neutrons are just neutral. Okay, so that should be hopefully relatively easy to remember. And obviously, if you're going to have some positive charge, you probably have some negative charges. So you're thinking, okay, I've got the positive, I've got the neutral. Where's the negative? And these things are electrons, which just kind of float around the nucleus. So negative and positive attract, so you kind of get the positive nucleus attracting all these electrons in. And what they do is they just kind of orbit in these shells like this, and they usually go in pairs. So we have a number of different electron shells, but the first shell always has 2 electrons at its capacity. Okay, so we can't get more electrons in than those 2 in that first shell. Now, what we actually have is for every proton, we have 1 electron. So we would need to do another 4 electrons because I've got 6 protons in my nucleus case. In the basic stuff about atoms. So what I've drawn here is a nucleus, and in the same thing as a cell, really. It's just the center thing which houses some important stuff. Except in this case, our nucleus of the atom is actually the thing which has things called protons and neutrons. So protons are positively charged and neutrons are just neutral. Okay, so that should be hopefully relatively easy to remember. And obviously, if you're going to have some positive charge, you probably have some negative charges. So you're thinking, okay, I've got the positive, I've got the neutral. Where's the negative? And these things are electrons, which just kind of float around the nucleus. So negative and positive attract, so you kind of get the positive nucleus attracting all these electrons in. And what they do is they just kind of orbit in these shells like this, and they usually go in pairs. So we have a number of different electron shells, but the first shell always has 2 electrons at its capacity. Okay, so we can't get more electrons in than those 2 in that first shell. Now, what we actually have is for every proton, we have 1 electron. So we would need to do another
Kind of daydreaming. Just have a little daydreaming break.
the first shell always has two electrons attracting all these electrons in. And what they do is they just kind of float around the nucleus. So negative and positive attract, so you kind of get the positive nucleus attracting all these electrons in. And what they do is they just kind of orbit in these shells like this, and they usually go in pairs. So we have a number of different electron shells, but the first shell always has 2 electrons at its capacity. Okay, so we can't get more electrons in than those 2 in that first shell. Now, what we actually have is for every proton, we have 1 electron. So we would need to do another. We can't get more electrons in. So we have a number of different electron shells, but the first shell always has 2 electrons at its capacity. Okay, so we can't get more electrons in than those 2 in that first shell. Now, what we actually have is for every proton, we have 1 electron. So we would need to do another 4 electrons because I've got 6 protons in my nucleus. So you see how I've gone to another shell and I've got 4 in this outer shell and 2 in this inner shell, so that totals 6, and I've got 6 protons in the nucleus. Now, you can have varying number of neutrons. So that's why I haven't discussed the neutrons yet. Usually it's 1 to 1. So usually you'd have 6 electrons, 6 protons, and 6 neutrons. However, the number of neutrons can change. So just keep that in mind, that this could be 5 or 7 or 8. It's always going to be around about that number, but it can change. Now, how we write this is we have some kind of letter which symbolizes our element. Then in the top left, we have this thing called the mass number. And in the bottom left, we have this thing called the atomic number. And then the top right, we have the charge. So we'll start with the atomic number. The atomic number is just the number of protons or electrons, because we always have the same number, but just think of it as the number of protons, okay? And this is the number that you will see written in the periodic table. So we have that big periodic table and it has all these numbers on it. That number is the atomic number. So if you know the atomic number of something, you know how many protons there are and therefore how many electrons there are as well. The mass number is the number of protons and neutrons. So what the mass number is actually how much that element will weigh. Now, the only things which have mass are the protons and the neutrons. So these are far bigger. What I've kind of drawn here with the electrons is just a letter, and that's to symbolize they're very, very small, far smaller than the protons and neutrons. And because they're so small, they have barely any weight. So the only things which contribute to the mass number is the number of protons and neutrons. So usually the mass number will just be double what the atomic number is. Remember that the mass number will just the only things which contribute to the mass number is the number of protons and neutrons. So usually the mass number will just be double what the atomic number is. Remember that the number of neutrons can vary. So the atomic number can't change, but your mass number could. And the last thing we have is this charge. So usually we have, as I said, we usually have the same number of electrons, same number of protons. However, in some cases we could lose an electron, or it's usually electrons, but I guess you could probably lose protons as well, or you could gain electrons as well. So this charge could be, positive or 2 plus or 2 minus or just minus, any of these variations. But based on that charge, you know how many electrons have been gained or lost, or protons. But I just keep it simple and just say electrons. So if it's 1 minus, it means it's gained an electron. If it's 1 plus, it means it's lost an electron. Because if we lose an electron, we're not going to lose that one, sorry, we should lose one of the outer shell. So if we lose that, we've still got our 6 protons in the nucleus, but now we only have 5 electrons. So that's why we would be positively charged. And just to finish this one off, this element would be carbon. So we'd draw a C there. Now, I think for the GAMSAT, you should just know a few different atomic numbers. And from that, you can kind of infer what the mass number is, right? So I would just know hydrogen, carbon, oxygen, and nitrogen. So I've done them in the wrong order. So this is them in order. Nitrogen, oxygen. And just know that this is 1, 6, seven, 8. So it's not the
6, 7.
perfect sequence, but it's relatively sequential. And I would just remember these numbers and then know that there are bigger things. So just know that chlorine, bromine, calcium, magnesium, potassium, all of these things, just be aware of them and know that they're bigger, but you don't need to know their exact atomic numbers. And the last thing I want you to be able to do is go from these numbers and realize how many valence electrons that atom has. So what a valence electron is how many electrons are in the outermost shell. So as I said, we have 2 electrons in that first shell. Then each subsequent ring can have 8 electrons. A little bit of variation to that, but that's good enough for us. We have 8, so this has 8 electrons in its outer shell. So you can kind of just keep going up, 2, then 8, then 8, then 8, then 8, and just keep filling those electron shells until you've run out of electrons. And then however many are in that outermost ring, that's how many valence electrons you have. So this is carbon because we've got 6 protons, and we know that our atomic number for carbon is 6. And you can see that we have 4 valence electrons here, because we have 1, 2, 3, 4 in the outer shell, and that's because there's 2 on the inner shell. So it's just about filling out the shells until you run out of electrons, and then however many are in the outermost shell, that's how many valence electrons it has. Okay, so we're going to get on to chemistry now, and we're just going to start, and then however many are in the outermost shell,
sodium. So sodium 11, 11 protons. No, sodium, sodium. 15? 11. It is 11. 11 protons. So it has what? 11 electrons. 2 plus 8 plus 2 is oxygen. So that's 8. 8. Alright. It is 8. 8. So it has 6. 3. Carbon. Carbon has 6. So okay, that's fine. I get that. Okay, I'm not going to do any
Next
more of
up, we're going
this
to
because I
talk
kind
about
of get the gist and it's a little bit boring. So I'm just going to move on and do some questions. Try and work through some questions. I'm going to be doing this one again. No, not the same question. I'm going to do 2, 3, and yeah, 2, 3, and then maybe move on to the other one. I'm just going to have a little bathroom break and maybe a cup of tea, and then I shall be back. Do this, ready? Crazy.
Here he is. Are you on some before your headset's off?
Oh, hello. I just got back from a little study break. Oh, good. I have constant problems with my microphone, so you just type in chat in the chat if you want. I'm not sure if you can hear me, my friend, or if you're only having problems with your mic or what the goes, but um, you can just type or whatever if you want to chat. Okay, cool. I'm just doing some practice questions at the moment. Before I was doing, I'm not sure if you've heard of a guy called, he's got a thing, free resources on Jim's GAMSAT. Yeah, that guy. He's pretty good. So I was just doing the skill, like the skills sort of development thing that he has, and now I'm doing the practice test A, the pink while I eat some yogurt. Um, I'm kind of going back and forth. Like I've done most of the resources. Like the thing I struggle with at the moment is, I mean, the skills part is like 50%, but then the like reasoning part as well. So at the moment I'll spend like a ridiculous amount of time on one question to kind of like step it out and make sure I know how to get the answer and why the answer is the answer, even if I know the answer, if that makes sense. So I'm kind of going back and forth between the ACE resources and then resources I find online and stuff like that. What about you? Are you using ACE resources? Now I also work on the yes, yeah, cool. Yeah, exactly. Um, yeah, for sure. Yeah, I'm on. So I just did the March sitting as well. That was my second. I've definitely got a few more sittings in me, for sure. Can relate, my friend. What's your study schedule like? So are you planning on doing the next sitting as well? Okay. What's Casper again? I've heard of it. I'm not sure what it is. Is that the Gold Coast Uni? Is that what they do? Bond or whatever. Oh, okay. Yeah. Right. Well, yeah. I'm here every day at from like 8 till 9. I do an hour. Um, I usually I stream it on Twitch. So I'm actually live on Twitch right now, so you can watch it there, or you can come in the Discord and hang out and study if you want, or just chat. It's up to you. But yeah, like you, I realized in the last sitting, um, how much I like don't know, and so I'm kind of motivated to spend the next 3 4 months just like getting the skills that I need, because I realized that some of the questions, I didn't actually have the skills that I needed to answer the question, and even if I could reason my way there, it wouldn't make a difference because I didn't have the skills. So just trying to build that foundation at the moment, and then also like spend a lot of time on the reasoning component as well. Yeah, great. Feel free. Feel free. Yeah, no worries. No worries. I'll see you tomorrow. Okay, that was fun. Um, yeah, like I said, so I'm just going to do the GAMSAT practice test A questions, 3 and 4 to 7 probably, and then we might basis of the information provided, which of the following is the most valid statement. So valid. So figure valid statement for figure 1. And the question, I guess we just define the answers, correct? Because it does see valid statement, correct, because it does not contradict table 1, okay? It's figure 1. Okay, so how do we find out if table 1 contradicts figure 1? It's defining the question, I guess. Text box here. One figure 1. Does table 1 contradict now? So I guess the table, like this blows my mind. So because I guess it's just knowledge that I didn't have before. But so you can grow a bacterium. So the Salmonella typhimurium, you can grow that on a medium. That bacterium cells. Try 1 to try 7, I guess. Supplemented with certain molecules. I just don't see action. I don't know how to find what would a contradiction look like. I think we'll just leave that because correct because a single enzyme catalyzes steps 3 and 4. That one out. Incorrect because it is not sufficient. Table 1. Correct because a single enzyme could not catalyze steps 3 and 4. I don't know. I think it's A. That's right. Question 2 is A. Yeah, fair enough. Okay, question 3. According to table 1 and figure 1, effect in a try 3 it could direct only. Sorry. According to table 1 and figure 1, the defect in try 3 directly affect only. Guess the defect is A in IGP. A is in 3. does not grow. Because it needs another supplement. So IGP grow. Yeah, I think it's 1. Step 1. It's 1. No, I've seen the answer though, it's not 1. It's C. Step 3. So I gotta figure out how to get there though. Maybe if I give A, that's fine. It doesn't grow. So to be indole glycerol phosphate to serine or indole. Oh, okay, if I give indole or tryptophan, it grows. Yeah, it's 3. Okay, that makes sense, I guess. Okay, we'll do question 4 to 7 and then we'll wrap it up. cleavage of which of the four tosin is facilitated? Yeah, which nonapeptide is cleaved by trypsin? Agripressin, oxytocin, octopressin, cephalotocin. Peptides. Okay. Okay. To sustain. So gives the name. Okay. Break apart or cleave a polypeptide. Polypeptide, is that a nonapeptide? That in laboratory enzymes such as trypsin and chymotrypsin are used. Trypsin carboxyl side of the amino acids, lysine and arginine. Okay, trypsin facilitates the cleavage of the peptide bonds carboxyl side of the acids. Okay, the carboxyl side. Carboxyl side. Carboxyl side. Carboxyl side. Acids, lysine and arginine. Okay. Arginine. Followed by proline. Okay. So we can go. Let's do. Let's do. Trypsin. Trypsin cleaves. Lysine. Arginine. Proline. Lysine. Trimotrypsin cleaves peptide bonds at the carboxyl side of the amino acids. Tyrosine. Okay, I don't even need that. So agripressin. Let's look at that. Sin. Agripressin. There's an arginine, followed directly by proline. Oxytocin. There's no lysine or arginine, so oxytocin's a no. Depressin. No, octopressin. Cephalotocin. Is that even cephalotocin? So there's an arginine. Cephalotocin, is that a depressin? Lysine. Wait, there is an arginine. It's followed by proline. So it's cephalotosin and maybe agripressin. Is that an option? B, but proline is there, but it's on the other side. Just think it's B. Do B. Let's do B. Or we'll lock in B. Amino acids could be isolated when chymotrypsin facilitates the cleavage of peptides agripressin. Equals. Okay, so chymotrypsin cleaves peptide bonds to the carboxyl side. Tyrosine, tryptophan. Tyrosine, tryptophan. acid could be isolated when chymotrypsin facilitates the cleavage of the known peptide agripressin. I didn't even know that. Oh, okay, I see. Tyrosine, tryptophan, phenol. Phenylalanine, no? A, let's do A. To the products when the cleavage of peptides cephalotocin is facilitated. Products when the cleavage of peptides cephalotocin. Products. Cephalotocin. D. 6 is D. 7. Okay, last one. Suppose that for easier and dipeptides. Trypsin, chymotrypsin, and other enzymes should be able to peptide bonds at the carboxyl side. Agripressin. if proline works there. That also works. It's not glutamate because there's either proline or cysteine. Oh no, it can't be cysteine because it's followed by proline, so it's alright. Let's check those answers and then we'll. Four B, got that. 5A, we got that. 6D, we got that. 7B, oops. Okay, so I've cleaved arginine. That's 3. Cleaved. Oh no, because if I'm cleaving, yeah, whoops. Yeah, that I can't cleave. I cleave proline. Yeah, then I've got 4 actually. That makes sense. Okay. Very good, very good. We'll call it here. Okay, that was a very good study session. Pretty long one, but that's okay. We'll be back tomorrow and we'll do some more practice questions, and then we'll probably get stuck, and then we'll have to go back to doing some other stuff like we'll do the skills and the chemistry and physics stuff and math stuff, which I struggle with. But overall, very good. And